This is Dr. Benjamin Shogan, who's joining us. He's did his undergrad at University of Rochester and med school at Rutgers. He did his internship and residency here at the University of Chicago, and then did his fellowship at the Mayo Clinic. He's a colorectal surgeon in the Department of Surgery, but also does translational research, which he's going to talk to us about today. So I'll turn it over to Dr. Shogan. Thank you.
Great, thank you. Thank you so much for the invite to come and present. I didn't really know what to talk about. A lot of my research is basic science and translational, so we're gonna get into that a little bit. But Dr. Golden had asked me just to sort of describe how I got into it, and so I think that I'm gonna just start from the beginning, and through this weaves kind of the story of when I was sitting in your guys' shoes, maybe a couple years before that, actually, to where I am now. And please feel free. I don't know how long this is going to be. My guess is about 30-35 minutes. But if some of the basic science is kind of heavy, and please stop me, or you can be interactive. You can write in the chat box. If someone writes in the chat box and I don't see it, then Dan, if you don't mind, just interrupt me and let me know.
Absolutely.
One thing, there's also some jokes in here, and because you guys are all muted, I can't hear you laugh. So it's sort of like making a joke to yourself in a room by yourself. So I guess you can either think that was
I chuckled at that one. So that
yeah, there we go. There we go. Zoom is going to have to somehow figure out how to make like a laugh track.
Oh, you got a ha 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 in the chat box also.
Yeah, perfect. There we go. Good. So I'm from New Jersey. This is where I grew up, right next to Princeton. And really, as long as I could remember, even when I was a little kid, I wanted to go into medicine. And I don't know why that was. I wrote in my essay that I wanted to save the world, but I've always wanted to go into medicine. And when I was in high school, some of you might know this show. This was like my favorite show, and all my friends knew I wanted to go into medicine, and people would call me John Carter, and I love this show, and I still love it today. I went to University of Rochester right out of high school, and in my pursuit to go to medical school as a junior, University of Rochester, I took the MCAT, and I did really bad, like so bad. I did so bad in verbal that it was almost like it was embarrassing. And about 6 months later, I applied to 13 medical schools. They all took my application fee and all rejected me in the same breath. And sort of my entire plan of what to do was really shaken up. I had a BS degree in microbiology because I was interested in microbiology, because I thought it would make me a good doctor because patients get sick. But certainly when I was choosing that degree, it wasn't because I wanted to use it for anything more than to get into medical school. I really had nothing to do. So as a senior in college, I started looking for jobs. And when you have a BS degree in science, you either go to medical school, you go to graduate school, or you become a lab technician. And so I became a lab technician in this guy's lab, Don Cohen, at Harvard Medical School. If you guys ever been there before, it's a beautiful, beautiful campus. Different from where the undergraduate is in Cambridge. It's in Longmeadow. It's right next to Fenway. And I spent 2 years working as a technician researching herpes simplex virus. Didn't know anything about it at the time, had no interest in basic science before I joined this lab, but I loved it. I really did fall in love with basic science, and I was able to kind of capitalize on my microbiology degree to be successful in this lab. Eventually took the MCAT again, did better, and then went back to Jersey, to Newark, to New Jersey Medical School. Graduated, and then did general surgery residency at University of Chicago, starting in 2009 and then finishing 2016. One thing that's unique about general surgery, especially at the big academic places, that maybe many of the students know or don't know, is we often take off 2 years to do some type of research. It's built into the program, and that research can be anything. It could be clinical. The American College of Surgeons headquarters is in Chicago, so a lot of folks go downtown for their research. But I was interested in basic science. I had done it before. I didn't want to make a career out of doing science, but I thought it would be a good fit for the 2 years. And so when I was on service as an intern, there was an attending named Dr. Alverdi, who, and I remember this conversation clearly, it was the end of my intern year, which is sort of when you start deciding what you want to do. And I heard that he studies bacteria. And he says, yeah, you know, I have this kind of crazy idea that bacteria may influence surgical outcomes. And this was, again, 2011. And it was right at the beginning of this kind of this boom in microbiome sciences. And again, I never would have gotten to this point if I did not get out of medical, if I didn't fail to get into medical school the first time. So I spent 2 years studying this, and this is a lead into what I study now. As you guys probably all know, colorectal surgery involves cutting out a piece of the pipe and reconnecting it. This is just an example. You know, your intestines is one long tube from your mouth to your anus, and things can happen. And when things happen, we can take out that disease segment and then tie the two ends together. It can be for diverticulitis or colorectal cancer, inflammatory bowel disease. The reconnection, which is here, that's called an anastomosis. You'll hear that term a lot in the next half an hour or so. And just like plumbing, you obviously want those connections to form a watertight seal. But about 10% of the time, sometimes more in high-risk patients, it can spring a leak. And when it springs a leak, it's a major, major problem. It's a thing that's on my mind for the first week after surgery every time. The patients can get very sick. They can be transferred to the intensive care unit because you can imagine what's leaking is stool. And obviously stool isn't meant to be outside of the intestine. Oftentimes they need to get an ostomy, and oftentimes that's permanent. And even those patients that do recover, they oftentimes have very poor bowel function afterwards just because of all the scar tissue. That's that anastomosis. So up till this point in 2011, before working with Dr. Alverdi, when leaks happened, it was pretty much because it was a technical issue. Either there was too much tension on the reconnection, or maybe the blood supply wasn't as good. But the idea that bacteria was involved in it all up to that point wasn't really discussed. And even looking at this graph, even though we have robotic surgery, or laparoscopic surgery, or advanced patient selection, the anastomotic leak rate, or the rate in which patients are leaking, was really been the same for decades before that. It really shouldn't have been surprising that the gut microbiome would affect healing. As we know now, and many of you probably know this too, is you have 100 trillion intestinal organisms. There's 10 times greater number of intestinal organisms than there are human cells. Just think about that. And there's almost 50 more bacterial genetic material as there is human material. So we're more bacteria than we are human. And we really think of ourselves as living in concert with bacteria. We can't live without it. They can't live without us. And what we know well now, I'm sorry about that, is that there's a whole bunch of different things that the gut bacteria play a role in or influence: cancer, asthma, inflammatory bowel disease, obesity. And every day there's another report. There's a link to depression, anxiety. There's the whole gut-mind axis. It's a defense against pathogens. It makes vitamins. And so, of course, you're tying two ends of the colon together. How does the bacteria not affect that? So to study that, and one thing that's really cool about basic science of translational labs is you get to do and especially in the surgical environment, is you get to have a surgical problem and then model it. The way that we do it is on mice or rats. And so we took a mouse, and again, as a year or two young resident, we created a model of an anastomosis as if the mouse had colon cancer. So we would cut out this piece of the intestine. These are pictures that I took from 2011. I would tie the two ends together. I would be pretending like I was the attending. And we would do that, and we would sacrifice the animal on day 6 and just see how well it healed. And this is the anastomosis. You can see the sutures there. And what we did is the first thing that we looked at is how does the bacteria change? on the anastomosis on post-op day 6 versus post-op day 0. And what we found is that there were major differences. And this was, at the time, you know, quite surprising. And the major difference that we found is this one bacteria called Enterococcus. And on the anastomosis, for one reason or another, Enterococcus had a 500-fold bloom on the anastomotic tissue compared to the tissue on post-op day 0. And this was an intriguing finding. We didn't understand exactly what this meant at the time. So what we hypothesized, and again, this is really a focus of our work and a lot of the work that we do in the Department of Surgery, is that you can have bacteria, and all bacteria aren't created equal. Different strains of Enterococcus function differently, just like different strains of human function differently as well. So what we hypothesized is that you have normal flora, and there's some type of stress, ischemia, radiation, malnutrition, you know, cancer, and that changes the type of bacteria. And then you get a proliferation of a pathogen. This pathogen can then lock onto that healing tissue and cause that tissue to leak. And this was our hypothesis. For about 2 years we studied this. I'm going to just show you a couple of experiments that we did, which I think they're kind of cool and very straightforward. Again, we do the anastomosis, and then we cause this host stress where we just cut some of the blood vessels to the tissue. And what we found is that half of those animals on post-op day 6, actually, they leaked, just like human patients do. We said human patients leak 10% of the time. We were able to do it in mice or in rats about 50% of the time. And then what we were able to do was we did all the different groups. We did animals that got an anastomosis versus not. We did animals that that got ischemia or not. And then we looked at the bacteria on post-op day 6 in all those different groups. And what we found is that the bacteria in the group that got the anastomosis and the group that got the ischemia was completely different on post-op day 6 than animals that got just an anastomosis or animals that got just the ischemia. So there was something about that environment, anastomosis and ischemia, that was changing this bacteria. And then what we did, what was cool, is we were able to phenotype that bacteria. And what we wanted to test was the bacteria's ability to degrade collagen. As you guys have probably learned, collagen is very important for wound healing. And so we thought if the bacteria is able to break down collagen, then maybe those bacteria are the ones that are breaking down that new connection. So we did that. And what we found is that the bacteria, only bacteria that had the ability to break down collagen was on those anastomoses that leaked. And even though you had Enterococcus that couldn't make that collagenase enzyme, and it was there, they didn't leak. And this was a cool finding. This was the kind of the beginning of figuring out how bacteria can be involved in something. So here, bacterial collagenase, the ability for bacteria to break down collagen, is what was always in the animals that leaked, which was never in the animals that healed. So then what we did, and this is actually one of the medical students that was in the lab at the time, actually one of the undergraduates, his name was Preston Lee, who then went to Dartmouth for medical school. He was an undergraduate. This was an undergraduate project. As he looked at all those bacterial communities recovered from all the different groups. And because we're talking about an entire community, we don't know exactly what strain is it. Is it Enterococcus or E. coli? Is it Pseudomonas? We didn't know. So what he did is we gave him a bunch of different bacterial communities, and on agar plates, he isolated and characterized all the individual species within all of those different communities. So now he had this entire library of different strains of bacteria. Then he screened all of them for their individual ability to degrade collagen. And what he found is when he did that, is that it was only those Enterococcus strains that had the ability to degrade collagen, whereas all the other strains he tested and isolated had no ability to degrade collagen. And up to this point, Enterococcus was really thought to be a commensal, really with no pathogenicity within the gut. And what he found is that when he stratified those Enterococcal strains, again, the same strain, but different phenotype, into those that leaked and didn't leak, again, it was always the Enterococcus strains that had that high collagenase phenotype was always associated with leaking animals, whereas the ones with low collagenase were always associated with healing or sham and operate animals. And Enterococcus is well suited to colonize this anastomotic tissue. As it turns out, it has receptors really for all of the different wound healing proteins that you have learned about or you will learn about. Fibrinogen and platelets come in, and fibronectin and collagen is laid down. Enterococcus uniquely has receptors on its outside membrane that can specifically bind to each one of these. So I guess in retrospect, it's not surprising that Enterococcus loves binding to the anastomosis. And then we did this experiment, and Preston helped me with this, which is a really cool experiment. We took animals and we gave it anastomosis, but no ischemia. So there was none of these animals leaked at all. And then we gave an enema of either high collagenase Enterococcus or low collagenase Enterococcus. And what we found, I'm going to skip over this part, is what we found is that when we gave the high collagenase Enterococcus, the animals leaked 100% of the time. And the only animals that I ever got to die whereas when this high collagenase Enterococcus was there. But when you have a low collagenase Enterococcus, even though it's Enterococcus, they didn't leak. They were completely healed. And with no bacteria, they didn't heal at all. So this was 2 years of work. And this was my surgical research fellowship. Again, working with undergraduates and medical students for part of it. You can see some of the names there, showing that some type of trauma changes the bacteria, and the bacteria can change phenotypes, and the phenotypes can bind to that anastomosis and cause it to leak. And this was interesting work at the time. I didn't think leaving this was in 2013 when I stopped. I went back to being a resident. I still had 3 more years of residency. I loved doing this stuff. I thought it was really exciting. We presented it kind of across the country. I never really thought that I would do it again. And when I left in 2016, I went to Mayo Clinic to do colon and rectal surgery fellowship. And about midway through your fellowship is when you start looking for jobs. And I interviewed places, and then what I call the bosses at the Department of Surgery, Jeff Matthews, who's the chairman, Neil Hyman, who is the section chief of colorectal surgery, and then John Alverdi, who was the PI of the work I just said, contacted me and says, "Hey, any interest in coming back and doing some more science?" And at the time, I loved it, but I didn't really think about doing it. And as it turns out, I joined faculty in 2017 as a researcher. I got my first, what we call, your real job. This is a real picture, September 1st, 2017. That's me. I don't know if they still do this, but at the time it was really big. When your kid goes to kindergarten, they put in, your first day at kindergarten, you put in what you like to eat or what you like to do. And it's fuzzy on purpose because it's not going to say there what I like to drink. And that's my daughter, and she was starting her first day of N3 down the street. So I joined as faculty, and based on the work I had done before, I said, I'm going to do a new twist on it. I'm a colorectal surgeon. Anastomotic leak is a problem, but cancer is probably a bigger problem. And as you guys probably know, colorectal cancer is the 3rd most diagnosed cancer in the United States. The 2020 estimates from the American Cancer Society say that there's going to be about 150,000 new cases diagnosed, a total. 2/3 of those are colon cancer, about 1/3 of them are rectal cancer. And overall, colon or rectal cancer is one of those cancers that it's pretty good to get. It's not bad like pancreas. It's not as good as thyroid. The outcomes are here. Localized, meaning it doesn't spread to the lymph nodes. Your 5-year survival rate's about 90%, which is pretty good. But once it starts spreading outside of that, outside of the colon, then your survival decreases quite significantly. And when it has metastasized, which is here by distant, either to liver or lung, your 5-year survival is quite bad, just like pancreas. And so this is quite a big problem. And as a colon, and I'm just going to go through kind of 2 patients that made us think about this new project that we focused on. And I will say, for any of those that end up going into research or translational research, especially on the surgery side, is one great way to think about patients is just going, or think about projects, is just going to the clinic and just seeing a patient and say, what about this? What about this doesn't make sense? Because it's really hard to be reading a literature, for me at least, and saying, you know, what don't we know? But when you go and see the patients and you use them as sort of like your experimental platform, you say, what about this? What about how am I treating this patient? What doesn't make sense? And then you go back and then you see if you can design a project around that. So this is a patient of mine, 67-year-old female. This is one of my first patients as an attending. She had bright red blood per rectum. Colonoscopy found a colon mass. It was confirmed as adenocarcinoma, which is the most common type of colon cancer. You always want to stage it with a CT, chest, abdomen, pelvis, and because you want to know if it's metastatic or not at that time. And if it isn't metastatic and it's isolated just to the colon, just like we talked about, the standard therapy is to resect that part and tie the 2 ends together. And this is a specimen. This is the colon. This is the mass. You can see it there. It's sort of opened up so that the pathologist can see it. And the final pathology revealed sort of the run-of-the-mill stage 2, not the best, not the worst, standard colon cancer. And when you have these patients, again, it's one of those cancers that when I see them, I say it's definitely not the worst, and you have a chance of surviving, a real chance of curing this. But then 1 year later, when she was undergoing her routine colonoscopy, we found cancer right at that reconnection, right? And we call that a local recurrence. And the framework, why, she asked what happened, and we say, well, there must have been cancer cells, even though this doesn't really make sense, but there must have been cancer cells at the edges that when you tie the 2 ends together, you get a new tumor there. And as bad it is telling someone they have cancer the first time, it is way worse telling them that you've gone through all this therapy, you cut it out, and then it comes back a year later. This is another patient, similar situation, 57-year-old female. She had rectal cancer, undergoes a standard therapy, which you guys will all learn about. Again, do a surgery in the hospital, goes home. And then 2 years later, we do a CT scan, and you can see there, that's a recurrence now in the liver. So we call this a distal recurrence because it spread distally from where the cancer was originally. Despite surgery being, and surgery is the primary treatment for cancer before, recurrence, but despite surgery being the primary treatment, just like for those patients, about 25% of patients will get a recurrence, either distally or locally. And unfortunately, in most patients that get a recurrence, they will end up dying. It's sort of like when they present with metastatic disease, whereas you don't really do surgery because it's spread, and instead they get chemotherapy, but they'll never actually be cured. And when you get a recurrence after surgery, again, the chance that they're alive in 5 years is somewhere between 25 and 50%. And this is the case even for low-grade tumors. So these are the lowest grade tumors. 4 is the worst, 1 is the best. And again, a stage 1 or stage 2 tumor, even these patients, 90% of them will be cured, but still 10 or 15% of the lowest grade tumors will have a recurrence after surgery. And it just didn't make sense to us. Like, why would that be? We cut out, if the tumor is in the middle, we cut out sections that are centimeters away from it. There must be something else going on. And so based on my background and the team I work with, background on the microbiome and the work that I presented previously, we wanted to look at how could those changes that we found that cause leak influence maybe cancer recurrence. And we know now, and many people know this now, is that bacteria has a strong effect on cancer pathogenesis to form cancer. So it can do all the things that you've learned about why cancer forms. It can cause inflammation, which causes cancer. It can produce reactive oxygen species, which, as we all know, is bad. But really, the role of bacteria in the development of cancer recurrence, which is my field, has really been largely unstudied. And so we developed a hypothesis based on the stuff that I did previously. You have a cancer, which is here. You do a surgery to cut it out. You have an anastomosis, which is what is here. Now, what we'll go into in a second is that it is known that even when you cut it out, there's some free floating cancer cells that are just floating around. And what we hypothesize is that those cancer cells can bind to that newly created connection, that newly created pipe, get influenced by the bacteria that is there, and cause either that local recurrence or that distal recurrence. And so the first thing we wanted to look at is, let's look at some of the risk factors for getting a recurrence, because maybe that would allow us to know what bacteria might be driving that. So we took animals and we fed them just a low-fat diet or a high-fat diet. And we know a high-fat diet is a strong risk factor for not only developing cancer in the beginning, but it is also a risk factor for developing a recurrence after surgery. And what we found is that in those animals that have a high-fat diet, they actually have more of that same bug that we talked about before, Enterococcus. We then did something really cool, is that we did surgery. We took animals, we started with 100 animals. We fed them either a low-fat diet or a high-fat diet. We did that anastomosis, where we cut the colon out, put it together. And then we sacrificed them every day for post-op day 1 to post-op day 24, so we could see dynamically how the microbiome changes every day. It was a bad day to be a mouse, that's for sure. And what these different colors show are different types of bacteria that was present either early on, post-op day 1, post-op day 2, post-op day 3, 4, 5, 6, blah, 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 comparing low fat versus high fat. And what is clearly different are these purple differences. And again, in the high fat diet, Enterococcus, even though both animals have gotten a reconnection or that anastomosis, Enterococcus really predominates in that first early time points, post-op day 2, post-op day 3, and only in high-fat animals. And again, like I spoke about before, a high-fat diet after surgery is a major risk factor for recurrence. So in this study of human patients who ate the most high-fat diet, which they did by quintile, had a 3.25 increased chance of getting a recurrence afterward compared to patients who were in the lowest quintile. And again, like I spoke about before, there are known to be these cancer cells that are floating around after you do surgery. And this was a paper a long time ago, decades ago, that showed that these cancer cells that were floating around in the lumen after surgery were actually viable and were able to regrow in animal models afterwards. And in this paper, they found that at the time of surgery, if you washed out the rectum, patients had a decreased recurrence rate. And what they hypothesized is that they had a decreased recurrence rate is because they were washing out all these viable tumor cells, but no one had put it together as to how this could actually form a recurrence. So we created another model in mice. And in mice, we gave mice a high-fat diet, like patients would get if they were, high risk for recurrence. We gave them antibiotics, just like you would get if you're a regular patient. We do the connection, like I said. Then we gave that an enema in some animals that of Enterococcus, that strain that we found to predominate in the high-fat patient and the high-fat animals. Then we introduced cancer cells as an enema to mimic those floating around, those cells that float around that are viable afterwards. And then we sacrifice them 3 weeks later, where we think it would be enough time for them to form a tumor or not. And what we found, strikingly, is that, and again, some of this stuff was done by 2 of my residents. There was a first and second year medical student involved in this as well. Is that we found in some of those animals, they form tumors just like human patients do, either locally, right at that reconnection, or we found tumors in their liver, which you can see here. These are the cells that we put in are fluorescent. So you can there's a way that you can look at the liver and look for fluorescence in the liver, and you can see that this is a tumor cell in the liver. So somehow it's getting from in here through the anastomosis and either making a tumor on the outside or making a tumor in the liver. We then found that the only times that the animals form tumors is, again, when the anastomosis is colonized the only time that they form tumors is when they're on a high-fat diet or when Enterococcus was present. When they're on a low-fat diet, they never form tumors. So there's something about a high-fat diet and Enterococcus being there that created an environment that allowed those cancer cells to migrate through. We then looked at the phenotype, just like we did in the previous studies, and we found that not only do you need a high-fat diet, and not only do you need Enterococcus, but the Enterococcus has to have a phenotype that can break down collagen. It's something about that phenotype, and we only found in the animals that form tumors, they only form tumors when a collagenase-producing bacteria was colonizing that anastomosis. And this is a cool picture because this kind of puts all of it together. This is the anastomosis there. Enterococcus is stained kind of that hot pink color there. That's Enterococcus. And this is the tumor. And you can see the Enterococcus going from the anastomosis and then migrating to the tumor, or vice versa, we're not sure. But you can see, and when you look within the tumor, Enterococcus is sitting on the tumor, influencing it. These are interesting studies, but what someone said is like, well, what if we just gave more antibiotics? Well, so we did that. So we gave vancomycin. What you guys will learn is vancomycin Enterococcus is very sensitive to vancomycin. And when we did that, to our surprise, tumors continued to be present. And we did knock down Enterococcus. But in its place, a different bacteria called Proteus blossomed. And Proteus now had an ability to break down collagen. And so it wasn't that it was specific to Enterococcus, but more specific to collagenase producing bacteria. And we weren't able to get rid of all the bacteria unless we use some type of advanced technology, something. You can grow mice without any germs at all. They don't have any bacteria, and we just haven't done that. And so what some of these kind of basic science experiments showed is that these tumor cells can bind through this anastomosis and cause local and distal recurrence. And their bacteria somehow plays a role because there's a very specific signature of bacteria for this to exist. But we wanted to bring it back to patients, right? Because at the end of the day, that's kind of what it's all about. And so, as many of you may or may not know, if you were undergoing a colon surgery, you would, I just have a few more minutes, you would meet the surgeon, and then if the surgery was on Tuesday, on Monday, the surgeon would tell you to drink a big jug of something called Golightly, so your colon is totally empty, and they would give you antibiotics so that your bowel was completely clean, or as clean as possible as bacteria. And that's kind of the standard preoperative bowel prep. And we started doing that at University of Chicago about 3 or 4 years ago for all patients. But before that, they weren't doing it. And the reason that we do it is to decrease infection. No one ever thought about it to decrease cancer recurrence. But because we've had a whole group of patients that hadn't had it and a whole group of patients that did have it, it provided us an opportunity to compare those two groups, which is what we did. And we looked at about 1,300 patients undergoing surgery for colorectal cancer. And we compared their outcome or their 5-year recurrence rate. And what we found is that when we matched them for all the different variables that may influence recurrence rate, that patients who had that decontamination had a significantly increased disease-free survival, or an increased or a decreased recurrence rate compared to patients that had no bowel prep at all. And this isn't reported yet. And this is quite an interesting finding, because it shows you that you can manipulate the bacteria potentially and change your outcomes after surgery. And where this is going is that is we wanted to look at the like I told you before, that the collagenase organisms we think is important. So we wanted to look in actually human patients to see if does that bowel prep that I just described, does that decrease the concentration of those collagen-lytic organisms. So we took human patients. This was also done by some students in the lab, and we looked at the colonization of those collagen-lytic or collagenase-producing organisms before bowel prep. So we actually have patients that are consented in the clinic, and then they go home with a box. They poop in the box, and then they send the poop to the lab. I'll tell you something, doesn't smell great after sitting in the box for a day or two, but we study it and we compare it to a sample that they give us at the day of surgery. So all we're looking at is how does that antibiotics decrease or increase or change the collagenase activity. And what we find, and when we look at all patients, it definitely decreases it. So bowel prep is good at decreasing those collagenase organisms. But remember, high-fat diet, obesity is a risk factor. So when we stratified patients by patients that are obese and not obese. And what we find is that patients that are not obese, the lowest risk, the bowel prep works great, decreases the collagenase activity very well. But in patients that are obese, those are the highest risk for recurrence. The bowel prep doesn't do anything for their concentration of collagenase activity. And we don't know how important this is. This is new. This is just recently published. But we think that there's something here that we might be able to mine and figure out what it is about those obese patients that is allowing it to hold on to those bacteria that may be their risk factor for recurrence. And then when we look at the different strains within the communities, we find that it's those same guys. The Enterococcus, we didn't talk about Pseudomonas, but Pseudomonas is there, and an organism called Proteus. So in summary, again, this is certainly raises more questions than answers, but what we've kind of investigated in the lab, sort of going both from patients to animals to the bench, is that tumor cells float around after surgery. They can be influenced by bacteria at the time of surgery that can cause recurrence. And this may lead to a kind of a unique opportunity as to how we can treat recurrence or prevent recurrence, or maybe even prevent tumors from forming in the first place. So just some take-home points. One is that intestinal microbes definitely play a role in development of colorectal primary disease, probably also recurrence. But I really wanted to use this to highlight at least my path. Again, not getting in and then being interested in microbes, and then using patients to sort of design a clinical project that now we're able to bounce between the clinic to an animal model and then back. And with that, I'm happy to answer any questions. This is my email. If anyone is ever interested in doing some research or chatting, I'm here. And thank you very much for the time.
Great, thank you so much. Are there any questions from the students?
I actually had a question about, you said that there's usually free-floating cancer cells after the resection. Are there any potential methods in which to potentially flush those out to prevent like the reattachment or like migration to distal areas, or
Yeah,
is
no,
that just like an inherent risk with resection?
no, that's an absolutely great question. And we do that when the cancer is on like the left side by the anus, where you can just flush it out. I mean, it's as easy as flushing out a pipe. You just put in some saline and you flush it out. And people have done that in randomized trials, and that does decrease the chance of recurrence, incidence of recurrence. But it's really hard to do it on cancers that are on the right side, that are the most common. The flush doesn't get all the way up there. It also is kind of cumbersome, and is a bit messy, and you're about to operate. So it isn't, it's not done often, but it's certainly done. The bowel prep that I was describing, where you get a big, jug of Golightly and the patient poops the whole day, that is a good way to flush it out. And again, we've shown that decreases recurrence, which is kind of cool. So it's a great question though.
Thank you.
I had a question, just kind of about your general career path. Is I'm curious what kinds of grant funding you might have had or currently have, and also kind of what's your current, like how much protected time do you have? I think as students often don't maybe understand how that works. So I'm curious kind of how you're set up in that regard.
Yeah, no, that's a good question. So so the way it works when you become faculty is, again, I only know about the Department of Surgery, so who knows if it's different. I'm sure it is different in other sections. Is that your time is sort of split. I know, Dan, you know this, but for the students, of course, your time is split, right? So you have a % of your time that is taking care of patients, which you call clinical time, and then you have administrative time, where, maybe you're the clerkship director or you're doing other administrative activities, and then you have research time. So my wife, who's a PED, she's sort of the most clinical that you can get, and she's about 85% clinical and about 15% admin. Me, I'm sort of on the other side. So I'm about 50% clinical and 50% protected time, which is quite a bit. I don't know in our department that is that protected to just do research. But I will say that protected time is something that's on paper, and it is something that's hard to do. So for students that are interested in research, or getting a career as someone doing research, you have to really be in the right environment, because as you guys will see, when you get into the clinics can be overwhelming, and the patients just keep coming. And so you need to be in an environment that is that you can have protected time and really actually be protected. In terms of funding, what most people will do is when you start, the department will pay for your for some of your research supplies. All this stuff costs money. And then you'll just start applying for grants. So there's a lot of grants available for junior faculty, a lot more than for senior faculty. So a lot at University of Chicago. So there's an institute called the Institute of Translational Medicine that I got a grant from. A lot of societies will have grants that you can apply for, and they're small. They're like 50 or 100,000. It doesn't sound that small, but MICER, all that stuff is expensive. And then as you go through those, you start applying for NIH grants that are more than 1 or 2 years, and those are obviously much bigger grants and such.
Cool. Any other questions from the students? I had one question maybe. What are your thoughts about the idea of circulating tumor cells, that are already in the bloodstream, potentially finding the wound healing matrix or milieu desirable, so to speak, as opposed to maybe free floating cells in the lumen?
Yeah, no question. And that's an area of like highly active investigation. And there's a couple studies that show that circulating tumor cells are a good predictor of recurrence, especially in colon cancer. I think that most of that data, or the mechanism that is thought to be for that, is that that's just a marker of cancer already being there. But we've tried to do the model in mice and then look in the blood for cancer cells. The problem is that the techniques to do that are really hard. Mice don't have a lot of you need a certain tumor mass to be able to detect it or not. But I think that a hypothesis is intriguing. You can see a situation where they get to the blood and then they bind onto something. The question is, how do they get there in the beginning? But that's an intriguing question.
Yeah, I remember, actually I mentioned to the students, Sam Hellman, who was the Dean of Pritzker 30 years ago, also a radiation oncologist. But I remember him talking at one of my morning conferences about some old study in women who'd had mastectomies and then died of other causes. And they did autopsies and looked in the mastectomy scars, and they could find like small tumor deposits that had never actually become recurrences. But I always found it interesting that if you have circulating cells and then you cause a basically surgical insult and you get all the wound healing growth factors and everything, it's kind of this prime location. Although then you'd expect people who had tumors and had wounds, other trauma would suddenly develop tumors there. So I don't know.
You can make a model where you do something in an animal.
I'm sure people have looked at this, and I know peripherally from conferences I've heard about it. But any other questions from the students, or?
I did have a question.
Yeah, go ahead.
Yeah, thanks for coming to talk to us, Dr. Shogan. I remember at the beginning of your talk, you mentioned that you weren't necessarily very interested in, wet lab or basic science research. And so I was curious about, because obviously you do a lot of kind of basic science stuff now. How did you have that change of heart? Was it just exposure or something else?
Yeah, I think it was, it was, I love basic science, or translational, whatever you want to call what we do. At the time, I was doing it both before medical school and when I was a surgical fellow. But the reason I didn't want to pursue it at the time to have a lab, honestly, was just because it was really hard. And I just didn't have an opportunity. I didn't think that there would be an opportunity to do that. I just, you go to 7 years of general surgery training and another year or two of fellowship, and then who wants to spend half of your time then, doing basic science, even though I loved it, especially if it was going to be so hard. And to what Dr. Golden said is, you need to find, if you're going to do it, you need to find a place where it is really supportive. So I was in a sort of unique situation where the bosses came up to me and said, "Hey, do you want to, continue some of the stuff that you're doing in an environment that was very supportive?" And that really sort of changed my view on it. And I'm so happy how that all happened. But your view definitely changes as you go through kind of your training. Everything kind of changes. So, but it's a good question. I never thought when I started as an intern, as a residency, that half of my time would be doing basic science, but it's some of the more fun stuff that I do, for sure.
Thank you.